I'd like to welcome you to our very nice surroundings, which is definitely a step up from last year. Hopefully you've benefited from the parking and the fact it's slightly better access. So I'm just going to start by introducing you to the clinical team. We have Thomas Barber here, who will be talking about prodromal or early Parkinson's in the RBD patients. We have Christine Lowe, who's just started, and we have Mark Kelly, who we actually didn't have to fund, but who spent 2 6-month blocks working with us and looking at impulsive compulsive behavior symptoms. We are very ably supported by administrative staff. Many of you may know Amandine and Jane from telephone and clinic visits. Marie Crabb is our new research nurse assistant or research assistant. We have Sam Avetz in the lab. We're fortunately now, after Sam has personally biobanked in excess of 25,000 samples, I hired a second person now to help him. And then we have Professor Ben Shlomo and Michael Lawton from Bristol, who are helping us with the statistical and epidemiological aspects of our cohort. And we have the Dendron nursing team. And many of you who've taken part may recognize some of our previous clinical research fellows that you may have met before. So just to recap about the cohort, can I just ask perhaps for you to put your hand up if you've actually taken part as somebody in the cohort, either with early Parkinson's, as a control, as somebody with RBD, or as a family member, possibly a sibling, mother or father. So just looking at that, I would say it's probably at least 70 to 80% of us here have had a link. As you may know, we stopped recruiting people with Parkinson's 2 years ago, and we reached our target of over 1,000 people with early Parkinson's. And if you were recruited in 2010, given that we now have extended our funding to 2020, you are one of the longest and most intensively followed up people with Parkinson's every 18 months in the whole world, I think it's safe to say. And many people also took part because their partner has Parkinson's as what we call a spousal control. We also had some members of the Garsington Book Club who all took part and were highly academic. And then we had our at-risk cohort. And the at-risk cohort is very important for us because we think that they represent, in particular those with rapid eye movement sleep behavior disorder, one of the earliest forms of Parkinson's, actually before it's even diagnosed. So obviously newer treatments, one could argue, may be best targeted at this group. In 2015, we were happy enough to get renewal of funding for another 5 years. However, there will be no OPDC-3 as such. And so we are looking at future funding to take us beyond 2020. And there is something on the back of the leaflet, the cohort participant newsletter, which does talk about personal donations, wills and legacies, if you were interested to consider that. One of the main issues, in fact, actually, has been of the 1,087 people we initially recruited, is actually following up people, and sadly 56 people, which is 5% of our total cohort, have sadly passed away since we first started this recruitment phase in September 2010. A number, 30, were reclassified following longitudinal review as not having Parkinson's. And we were then left with just over 1,000 people eligible for follow-up every 18 months. And what you can see here, and that I'm very proud of, is about 84% of these eligible people are now being followed up through clinic, and a small proportion through telephone follow-up. What are the cohort objectives? It's very important to keep going back to why we set this up in the first place. I think the first thing is we're interested in predicting Parkinson's at its earliest stage, and you will be hearing a lot more about this through the work of Tom Barber and Christine Lowe, focusing on sleep and prodromal RBD. We're also interested in people who have different phenotypes at the time of diagnosis, and that's basically everybody here with Parkinson's, because everybody behaves differently, firstly in the initial symptoms, secondly in their response to medication, and importantly in how they progress. And that difference we call heterogeneity, and we're very interested in what we call stratifying Parkinson's to develop meaningful pragmatic markers in a clinic that could be used to tell an individual at the time of diagnosis where they might expect to be at 5 and 10 years into their Parkinson's journey. We're interested in correlating what we're finding in our so-called biomarkers, and these could be simple clinic-based tests, a blood-based test, a spinal fluid test, or an imaging test. And I've given you here an example of some of the cutting-edge imaging we're doing involving MRI, the picture on the left, and Dr. Barber now doing a functional SPECT, or single photon emission tomography, which is being done now in 50 people with RBD and parallel MRI. So we want to understand the biology of what's happening in the brain better and to validate that. But what I'm really just going to spend the next 10 minutes talking on is novel therapies, which really is what we're focusing on in 2015 to 20, and in particular in the next 3 years. So to update you really just on a few interesting studies that are on the immediate horizon, I'm going to cover immune therapy treatments, in particular alpha-synuclein antibody studies, which will be starting very soon in early Parkinson's, the PDStat trial, which aims to reduce high cholesterol and vascular risk and impact positively, therefore, on Parkinson's. I'm also going to be talking about Exenatide and type 2 diabetes treatments and where OPDC discovery may be coming in. And this is highly topical because of a very exciting paper that's just been published in The Lancet in the last month. And I'm also going to be talking about lifestyle exercise modifications, which will also reduce vascular risk, and how we can actually educate patients to self-manage their Parkinson's through the First Steps course, which was piloted in Oxford, is now being rolled out in Hampshire, and in fact has now been taken on by Parkinson's UK to be rolled out nationally. To be covered later by separate speakers will be treatment trials in gait freezing for Parkinson's, and in particular a device called Gait Thaw by James Cantley down in the lab at DPAG, sleep modification for Parkinson's, and drug and target discovery. So just starting with the alpha-synuclein antibody trial in early Parkinson's, or SPARC-PD. This is funded by Biogen and Quintiles. It is a multi-center, randomized, double-blind, placebo-controlled study. And this really is targeting alpha-synuclein, which is the misfolded protein that somehow deposits in the neurons, the brain cells of people with Parkinson's, and ultimately clogs up that cell functioning and leads to it dying. So this is really exciting because it's the first of its type that would try to give somebody an antibody through a drip, a bit like monthly cancer chemotherapy, hope that that drug will cross into the blood-brain barrier and the brain, grab hold of that misfolded alpha-synuclein protein, and ideally clear it from the brain to rescue brain cells. Now, how does this link to OPDC discovery? I've already pointed out the lady in the hat, Sally Cowley, and she has developed a very interesting way to study inflammatory cells called microglia in the brain, and actually of generating them from skin biopsies that we've taken from you here. And she's going to be discussing some of that cutting-edge research this afternoon. Secondly, vascular risk. What do I mean by vascular risk? I'm really talking about overweight, high blood pressure, high cholesterol, type 2 more than type 1 diabetes in people of the age that get Parkinson's, and also heart problems like angina and stroke. We are increasingly recognizing that if you have Parkinson's and these so-called vascular risk factors, the two egg each other on. And if you have several vascular risk factors, your Parkinson's is actually worse. You are less likely to respond to levodopa and other treatments. And actually GPs and clinicians are under-treating people's because they're saying, oh, it's just the Parkinson's, we don't need to worry about the blood pressure or the cholesterol. And the reason I can say that with some certainty is because of real-life data from our cohort. So we were actually able, in this paper on the right, to study nearly 3,000 people, both from Oxford discovery and tracking, and we were able to look at your vascular risk and see how that impacted on progression of Parkinson's. And we were able to show that, firstly, people who have Parkinson's are undertreated in terms of vascular risk, and secondly, if you have Parkinson's and vascular risk, your Parkinson's does not so well. So this, in fact, not necessarily our study particularly, led to what we call the PDStat trial. So could I just have a quick show of hands if you've taken part or are taking part in PDStat? So Francesco is the lead nurse at the back who's been leading. I see 2 hands. Okay, so PDStat is led by Camille Carroll at Plymouth Hospital, a neurologist like me. And the main question is, does simvastatin slow or halt the progression of Parkinson's through this vascular risk modification I've just been talking about? And so patients with moderately severe Parkinson's are randomized to receive either simvastatin or the dummy if they have what are called vascular risk factors that would justify treating the high cholesterol. They are followed up over 2 years, and recruitment began in March 16 from 23 hospitals, including Oxford, with the aim of recruiting 200 Parkinson's subjects. Now, I believe that Oxford is one of the top recruiting sites. How many have we recruited? So 17 people just from Oxford, which is great because, again, this trial has a lot of inclusion and exclusion criteria. That means we might be screening 5 people to find only 1 that's roughly eligible to consider taking part. The second vascular risk study I'd like to talk about is Exenatide, and Exenatide is part of a group of drugs called the glucagon-like peptide 1 agonists. Now, Tom Fultony, who many of you might know, and Professor John Todd, who you may not know. Professor John Todd is actually moved to Oxford to the Wellcome Trust Genetics Centre, and is one of the leading experts in genetic causes of type 1 and type 2 diabetes. Dr. Fultony has just published this paper in The Lancet after an initial study which found that in 60 patients with early Parkinson's randomized to either get Exenatide or placebo, they actually improved. They got better, the people treated with Exenatide, in their Parkinson's progression over 1 to 2 years, whereas the people with placebo got worse. Now, this is really significant because it's the first placebo-controlled study in humans to show clear benefit with Exenatide. Now, how might Exenatide work, and why on earth could diabetes, in particular type 2 diabetes, which Exenatide is used to treat, be relevant to Parkinson's? Well, firstly, we've known for some years that if, in fact, you have type 2 diabetes, which is the one more common in later onset of life, your chances of getting Parkinson's has increased by 40%. So we've known that there's an epidemiological link for some time. The pancreas, which contains beta islet cells which produce insulin, which is deficient or reduced in diabetes, is also very similar in some ways to the brain organ. In diabetes, the cells are attacked through autoimmune, inflammatory, or oxidative stress, and they die. Now, we can treat type 1 diabetes through giving insulin, but type 2 diabetes is often treated with glucagon-like peptide 1, which are given and improve insulin production and also insulin release. But what's very, very interesting is that these drugs have been shown to slow down Parkinson's progression in animal models, both MPTP-treated mice and also other genetic models of Parkinson's. But why might that be? The argument is that these drugs cross into the blood and they may rescue the brain cells from oxidative stress, mitochondrial dysfunction, and they may also improve cellular regenesis in the brain, very similar to their effects in the body. So what we've interestingly found, just by looking in our cohort of 1,500 people, is that Parkinson's, and this is just where people have said, I've got diabetes, is actually much higher than in controls. So we're seeing diabetes in about 9% of our Parkinson population and in 5% of our age and gender-matched control population. And in the prodromal Parkinson group, the RBD, we're seeing it in 16%. Now, many people here might actually have diabetes but don't yet know it, because it's a very silent thing. And what we are currently doing is working with Professor Todd and Dr. Fultony to get funding to check all the blood tests that you've given us for diabetes using an HbA1c marker, and then in the future to work together to potentially see whether if we treat somebody with Parkinson's and diabetes with Exenatide or an equivalent, they do better than somebody with standard treatment for their diabetes. And also the Exenatide trial that Dr. Fultony did actually didn't treat you if you had PD and diabetes. So actually the real money might be in treating people with Parkinson's and diabetes, or just Parkinson's without, but we don't yet know that information. So that's an important area of future study for the cohort. And then I'm very proud, as are many people here, of First Steps, which is a course for newly diagnosed individuals which was piloted and thought of by local people with Parkinson's in the group, and it's administered by people with Parkinson's. And based on the success of pilot work in Oxfordshire, First Steps, we were then able to get a grant from the Parkinson's Excellence Network to actually now roll this out in Hampshire with Helen Roberts, who is a gerontology PD specialist, and we will then be looking at the effectiveness of rolling this out in a different region, and we will be looking at whether information to people at diagnosis on healthy living, exercise and diet does potentially improve Parkinson's later on. So just to finish my talk in summary, we are about to start novel immunotherapy trials very soon for Parkinson's in Oxfordshire using monoclonal antibody infusions. And I believe that OPDC Discovery can comment on these underlying disease processes through the work of stem cells and microglia. Secondly, reducing vascular risk, and in particular obesity, high cholesterol diet, is possible through lifestyle, through exercise and medication. And we have here also Michele Hu, who's one of the community physios who's worked with us very closely to bed in exercise programs for recently diagnosed. And this could be a huge step in slowing down Parkinson's in the future. And lastly, I think you can be proud of your pioneering First Steps program, which has actually been designed and delivered by many local people with Parkinson's here, and has proved to be a great success. So thank you very much.